Welcome everyone to this Wednesday seminar. In the spirit of reconciliation, I acknowledge the traditional custodians of country throughout Melbourne and their connections to land, sea and community. Let me pay our respects to their elders and present past and present, and extend that respect to all Aboriginal and Torres Strait Islander peoples today. So it's a real pleasure for me to introduce Julie Illo, who is our speaker today, and Julie is currently the Insectary and Project Manager in the Division of Infectious Diseases and Immune Defence. Just a bit of background on Julie. She did her PhD at Edinburgh University in 1998, not so long ago, where she developed her long interest in malaria and diseases of resource-poor countries. She received a Wellcome Trust postdoctoral fellowship and came to WEHI in 2002 to my laboratory. The Wellcome Trust fellowship is very prestigious and also back then very lucrative, and I can remember I was extremely jealous because she was actually paid a lot more money than I was. But after completing a successful postdoc, Julie decided to stay in Australia and moved to the laboratory of Stephen Rogerson at the University of Melbourne, after which we were lucky enough to attract her back to WEHI. So Julie, at that time, was appointed as project manager for our Gates-funded project in collaboration with Stephen Capper at Seattle Biomedical Institute and also the Walter Reed Army Institute. And this project was to develop a live genetically attenuated malaria vaccine. Julie's role was to oversee the development of a good manufacturing practice facility at Bundoora to construct genetically attenuated parasites for human use in human clinical trials. Those of you who know what's involved in manufacturing GMP products will understand and the regulations involved in this. And Julie, along with her team out at Bundoora, did an outstanding job and successfully developed a number of genetically attenuated parasites that are covered by a patent and also have been in a phase 1 human clinical trial at Walter Reed Army Institute in the USA. And one has now completed phase 2 clinical trials in the USA as a vaccine against malaria. Julie has now taken on the role of manager of our insectary facility and is also project manager of our more recent vaccine project in collaboration with a company in the USA called Leidos and also USAID. It is this project that she'll be discussing today. Julie will discuss the current state and development of malaria vaccines and why it has taken so long to deliver on this urgent public intervention. She will discuss the results of our latest collaboration with Leidos in using our understanding of the biology to select vaccine targets. So Julie, looking forward to your talk. Thank you.
Well, thanks, Alan. Can everyone see my screen? Well, Alan, can you see my screen?
I can see your screen, yes.
Okay, great. Thanks very much. So I would also like to acknowledge the First Nations custodianship of the lands across Australia and pay my respect to their elders. So I'd like to move on to talking about malaria vaccines. So there's been a long history of interest at WEHI in developing a vaccine against malaria, and this started back in the 70s with the establishment of the parasitology unit here. The first antigens, the first Plasmodium antigens were cloned in the early 80s. And here's this young dude who's looking quite pleased with himself after having solved the sequence of one of those early antigens. So that work, the trajectory of those first antigen cloning experiments led directly into Alan's work in his own lab with the publication of the first gene deletion in Plasmodium falciparum. And that's when I first heard about Alan after that Cell paper was published. And I decided then that I wanted to come and work here. So in the 2000s, there was a huge amount of progress after the publication of the Plasmodium falciparum genome. And at that time, Louis Schofield had developed a GPI vaccine, which was looking really promising in proof of concept studies. There was a study which underwent phase 2 clinical trials in Papua New Guinea that involved work from Robin Anders and Graham Brown. And our Common Lab anti-invasion vaccine work began in earnest towards the end of the 2000s. And as Alan said, we've been involved in the development of several genetically attenuated parasites, but I'm not talking about them today. I'm going to focus on our work on an anti-invasion vaccine, and that work started late 2017. So I'll move on here. So first, an introduction to malaria. There are 5 species of Plasmodium parasites that cause malaria. And Plasmodium is a single-celled eukaryotic organism that goes through a number of distinct developmental stages. And there's around 5,500 genes in the Plasmodium falciparum genome, so it's very complex. By far, the most important species in terms of morbidity or virulence in humans is caused by Plasmodium falciparum, and that's the species that I'm going to concentrate on today for our vaccine work. So I would say that probably the biggest challenge in making a vaccine against malaria is this complexity. So when a person is bitten by an infected mosquito, it injects around 10 to 100 cells called sporozoites directly into the skin of its host. So these sporozoites migrate through the skin into the bloodstream and make their way into the liver. So over the course of an hour, this sporozoite form finds its way into the liver, where it goes under a huge expansion in numbers in a process called schizogony. So after about a week, these daughter merozoites formed by this step of multiplication are released into the bloodstream. And this is where the biggest expansion of parasite numbers occurs. So every 48 hours, the parasite undergoes this cycle of invasion and replication, this schizogonic cycle within an infected red cell. And the release of these merozoites into the bloodstream is concurrent with the onset of the symptoms of malaria. So it's this stage that we're going to concentrate on in terms of the development of a vaccine against the blood stage of malaria. So at some point during this cycle, there's a developmental switch that happens, and the parasites then commit to a sexual development pathway. And the development of these fully mature sexual forms takes around 2 weeks in Plasmodium falciparum, so that when a mosquito takes a blood meal, these egress from the infected erythrocyte inside the gut of the mosquito. They form male and female zygotes that fertilize and form a zygote, which then invades the midgut of the mosquito and starts again with another huge expansion, asexual expansion of parasite numbers. So one aspect of this complexity of this life cycle are these multiple different stages that seem to have very few antigens in common. And this is important for vaccine development, because an immune response that develops against, say, the sporozoite form is not cross-protective against the forms in the bloodstream. And the second point that we have to take into consideration when looking at parasite stages for targeting for a vaccine is that for the most part, these parasites are intracellular. And so they're immunologically silent. And the parasite has evolved all sorts of really cool strategies to avoid immune detection. So I put this slide in as a reminder that until actually very recently, malaria had a much bigger global footprint than it does today. And even in Australia, malaria was not eliminated until the 1970s. So it's possible that with enough resources and enough will, malaria can be eliminated. And in fact, the last 20 years since 2000 have seen tremendous progress in reducing the global burden of malaria across many of the regions that were endemic in the past. And this interactive map shows that the decline in malaria transmission from between 2000 and 2017. So the exception to the areas that have made huge gains in conquering malaria actually occur here in sub-Saharan Africa. And so this region has an enormous case incidence and malaria-associated mortality. And if we remember that the majority of malaria deaths happen in kids under 5 years old and also in pregnant women, this is a really big problem. So, you know, since 2000, there's actually been a 60% drop in global malaria mortality rates, which is really incredible progress. And what made that progress possible? Well, basically money. A huge expansion in financing thanks to the Global Fund, and an increased level of political commitment to making this happen. So it takes a really multinational approach so that countries can get together and come together to discuss malaria control strategies. But actually, how did this happen? Well, it was by the massive rollout of new medicines, by rolling out insecticidal treated bed nets, and by rapid diagnostic tests. So a substantial scale up of these core interventions. So why do we need a malaria vaccine if so much progress is made with the tools that we currently have? So in low-income countries, malaria is still the 6th leading cause of death after respiratory and diarrheal diseases, cardiovascular, HIV, and stroke. And since 2016, the malaria case numbers are on the rise again. And so that these gains in malaria control that were made since 2000 that are seriously under threat now due to the emergence of drug resistance in the parasite and the emergence of insecticide resistance to the mosquito vectors. So every 2 minutes, there's a child somewhere in Africa dying of malaria. And the tools that we currently have are not enough to control, never mind eliminate malaria. So is there already a malaria vaccine? Well, there is, and it's called RTSS. And it targets these sporozoite stages that are injected by the mosquito. And if you remember, an immune response targeting this stage does not cross-protect against malaria in the blood. So there's been an enormous investment into this vaccine over the past 30 years. Phase 3 trials across Africa involving in excess of 16,000 young children had disappointing vaccine efficacy, which appeared to decline fairly rapidly. So that efficacy in the early days of about 45% in young children and about 27% in infants declined to around 16% and under 8% in the smallest children. So obviously that's not going to be enough to add much to the toolkit to eliminate malaria. And it was decided that a 4th dose should be included in the implementation program of RTSS, which was administered 18 months after the initial course to see if that would improve the vaccine efficacy over time. So the target of RTSS is CSP, known as the circumsporozoite protein that was identified early in the 80s. And then there was a 20-year clinical development program in which the Walter Reed Labs partnered with GSK. And during that time, there were at least 6 GMP grade vaccines that were pulled because of poor efficacy. However, the GSK developed the adjuvant known as AS01, and that went through to phase 3 trials in 2007. And it was at that point that Bill Gates and his mates from the Global Fund decided to put malaria eradication firmly back on the agenda. So in 2009, these huge phase 3 trials that were funded by the Gates Foundation started in various countries across Africa. And where we're at now is these phase 4 post-implementation phase, where they're trying out this 4th fractional dose. So if we just compare this 40-year project with the COVID vaccine development, we can see that in the past year since the COVID, the virus SARS-CoV was identified and the sequence was published, we can see that there's been at least 150 vaccines in clinical development. And several of these are already in phase 3 clinical trials. And these are just a few of those promising candidates. So, you know, where there's a will and where there's money, there is a way. And actually the WHO are coordinating this massive response for 2 billion doses of a successful COVID vaccine to be delivered by the end of next year. So back to malaria, where are we now with the malaria vaccine? So there is a global technical strategy for an improved malaria vaccine, and that has called for the scientists and developers to basically develop a next generation vaccine that shows at least a 75% efficacy over a 2-year period. And that where we come in is that should include new strategies to target the blood stage or the transmission stages of malaria. And actually, there's been this recent paper published which has looked at the number of vaccines registered in the clinical trial registry. And since 2000, there have been 145 malaria vaccines registered in this registry. But interestingly, only 8 have targeted the blood stage since 2010. So as a lab, the Cowman Lab's mission has really been to better understand the function of parasite proteins to inform better vaccine development and for drug design. And we've really focused on this blood stage of the parasite for a number of reasons. Firstly, that it is the stage of the life cycle that causes all the clinical symptoms. Secondly, that it's easy to grow in vitro. All you need is a regular source of red blood cells, which we get delivered from the Red Cross every couple of weeks. And also it can be genetically manipulated. And so this movie here shows an actual live image of a malaria merozoite attaching to and invading a red blood cell. So as you can see, this process is extremely quick. And so although the merozoite is the only stage that's extracellular during the whole 48-hour blood cycle, it happens very quickly and the immune system has to actually find and attack this free-living merozoite stage. So it's still quite a challenge. So for the past 20 years, the Cowman Lab's been really interested in looking at the host parasite interactions that drive blood cell invasion. And merozoite invasion is a very rapid and stepwise process that involves multiple parasite ligand and erythrocyte surface receptors. So when the merozoites egress from the mother cell, they have to quickly locate and invade a new cell. And this whole process, as you saw in the movie, happens within the span of like a minute. And so it starts with this weak attachment phase to the red blood cell surface, followed by this interaction with the erythrocyte membrane that causes these deformations in the membrane of the erythrocyte. And so there's this kind of softening of the erythrocyte membrane, and then the parasite seems to wrap this membrane around itself, and all the proteins that are involved in the invasion process are housed in these subcellular organelles known as the rhopteries and the micronemes. So it has to pull its apical end round so that it's in contact with the erythrocyte membrane. At this point, there's a commitment to invasion and a strong attachment occurs. And we know at that point that there's a calcium signal between the merozoite and the erythrocyte. And then invasion proper can proceed. With the parasite pulling the red cell around itself and resealing of the erythrocyte membrane. And there's been a lot of work by our group and other groups to identify the key interactions that drive this process. So we created a series of genetic knockouts to try to characterize these critical interactions. And it's these superfamilies called the EBAs and RHs that are the main players in this interaction. They play an essential role in receptor binding and downstream signaling pathways. But individual members of these superfamilies can be knocked out. And it appears that there's multiple compensatory pathways. So each parasite ligand binds to a specific erythrocyte surface antigen. And there are multiple surface antigens on erythrocytes, as you know. Red blood cell antigens are extremely diverse. So it's believed that the evolution of these different ligands have allowed Plasmodium falciparum to inhabit a much bigger niche in the human red blood cell population. So the more essential pathway that we were able to identify is shown here on the right, is the RH5 pathway. So RH5 binds to an erythrocyte receptor called basogen, or CD147. And we had tried many times to knock out PFRH5 in various different Plasmodium strains, and both we and in another lab were unable to do this. So it looked like this was completely refractory to genetic deletion, meaning that it's essential for this invasion process. So we believed that actually blocking this interaction with antibodies would be a good way to target parasite invasion with vaccine development. And our friends at Oxford University, led by the group of Simon Draper, have also been working up this antigen as a malaria vaccine candidate, and they've really led the way in establishing preclinical models and clinical trials around this antigen candidate. So it turns out that RH5, although RH5 binds to basogen, it doesn't do it on its own. It's part of a complex with at least 2 other proteins. And that includes the RH5 interacting protein, RIPR, and CYRPA, which is the cysteine-rich protective antigen. And these 2 proteins also can't be knocked out, so they're also essential. So this whole complex is critical for parasite invasion of red blood cells. So how do we go about studying the function of essential proteins when we can't knock them out? Our lab took 2 parallel approaches. In the first case, we used CRISPR to create conditional knockdowns where you can control to some extent the timing of the gene deletion. And secondly, we undertook a program of recombinant protein expression to investigate these interactions. So I just want to acknowledge the work here of Jennifer Bowles, Jenny Thompson, Chavi Sisquella, and Alan Yap, and all their other collaborators on this massive study, where they knocked out or conditionally knocked down RIPR, SIRPA, and also AMA1, which is another essential protein in Plasmodium falciparum invasion. So if you grow these RIPR knockdowns, in this case, with wild-type parasites in the same dish, you can see that there's a severe growth defect when RIPR is knocked out. So the line in black here shows the growth increase in parasite numbers from the wild-type parasites compared with the RIPR knockdown parasites. So there seems to be a massive defect in the ability of these parasites to compete with wild-type parasites. And secondly, if you look actually just at the RIPR knockdown parasites and you follow the invasion events over the subsequent cycles, life cycles events, we can see that there's reduced numbers of invasion events over the subsequent cycles as the amount of RIPR in these cells is declining. So looking at those parasites under the microscope now, you can use time-lapse imaging to see the effect that a knockdown would have on the ability of a merozoite to invade a red blood cell. So I'll just wind this one back. If you concentrate on this cell that's highlighted here, you can see the merozoite. It's released from its mother cell, and within seconds, it's gone in. And there's this echinocytosis, which is a marker for the resealing of the erythrocyte membrane after invasion has happened. Contrast that with a RIPR deficient cell. So we've got these merozoites egressing here, and they can form these tight junctions or these tight associations with the erythrocyte membrane, and the merozoite is really pulling the erythrocyte membrane around itself, but it just can't get in. So that shows that the loss of RIPR is severely affecting the merozoite's ability to invade the red blood cell. And we know that calcium is important in this process. And these cells have been loaded up with a calcium-sensitive dye called fluo4. And if I can just pull this one back again, there's a little calcium spike in the merozoite just before the invasion happens. And then this is transmitted into the erythrocyte, and then invasion happens. So it happens, this calcium spike seems to immediately precede the invasion event. So what Jennifer did was to try to determine the role of RIPR, SIRPA, RH5 complex in this calcium signaling event and invasion. So if you look in normal invasion, there's a calcium flux that happens in two distinct phases. So in black here, we've got the calcium spike in the merozoite just before the invasion event, and also in brown is the calcium levels rising inside the red blood cell. However, when you knock down either RIPR or SIRPA, you can see this spike happening in the merozoite, and it's elevated, it remains elevated throughout the time course of this experiment, and there's no subsequent calcium spike in the erythrocyte. So it appears that the calcium flux into the red cell is dependent upon this RH5 interaction, or RIPR, SIRPA, RH5 interaction. And in fact, you can block this by the addition of soluble basogen, which is the receptor on the red cell. So also with anti-basogen monoclonal antibodies, you can block it with anti-RH5 antibodies to some degree, and also with antibodies against RIPR. But it's specific because if you use a PFEMA1 knockdown parasite, you don't see this effect on calcium signaling. So this is good evidence that the RH5 interaction with basogen depends on these other two proteins, and that initiates the calcium signal into the merozoite, from the merozoite into the red blood cell. And some recent work that Kelly Rogers' lab, Kelly actually showed in her recent seminar, using lattice light sheet microscopy, has shown this similar event, so that the merozoite comes along, there's a little calcium spike shown in yellow, and then you get invasion right into the red blood cell. So to summarize that part of the talk, I've told you that RIPR and SIRPA form a complex with RH5 during invasion, and that conditional knockdowns of RIPR and SIRPA block merozoite invasion, and that there's a co-localization of RIPR, RH5, and SIRPA that are associated with a calcium spike and invasion of the red blood cell. So the other track that we took in order to understand the function of these essential proteins was to undertake a program of recombinant protein expression for structural investigation. And this has been a huge amount of work done by our lab and other labs to try to express active and conformationally intact forms of these proteins, which in the case of RH5 and RIPR has proven extremely difficult and time consuming. So this one slide shows a few of the papers that have come out from this work, and in the last 10 years, and these are just a few, there's others as well from other groups. But I just wanted to point out and to acknowledge the work of Lin Chen, who discovered the partner of RH5, which was the RIPR protein, and also for her work on solving the crystal structure of RH5 and SIRPA. And also our collaborators and colleagues in Oxford, who solved the crystal structure of RH5 with its receptor basogen, and also with neutralizing antibodies, monoclonal antibodies raised against it. And then, of course, we published this paper last year in Nature. Wilson solved the cryo-EM structure of the ternary complex of RH5, SIRPA, and RIPR. So once we were actually able to get full-length RIPR, we were able to make this complex. And Wilson identified that the complex does in fact bind through RH5 interacting with bathogen through this sort of low affinity interaction. And then there appears to be some kind of conformational change in RH5, which allows the disengagement of SERPA. And this is followed by a much tighter affinity interaction with the erythrocyte membrane. And by electron microscopy, it looks like there's a big aggregation of RIPR and RH5 in the erythrocyte membrane. And we are hypothesizing that this is linked to the formation of a pore, perhaps through which calcium can flow. So you can raise antibodies against these recombinant proteins and purify these antibodies from serum. And then you can incubate these antibodies with parasites in vitro in like a 96 well plate format. So when you do that, and you have antibodies that inhibit merozoite invasion erythrocytes, we can use this growth inhibition assay to basically determine the effect of these antibodies on the ability of parasites to invade. So that in the absence of antibody, we see no growth inhibition, and in the presence of inhibitory antibody, we see a lot of growth inhibition. And so in that way, we can calculate the effect that different antibodies have on the replication or the invasion of merozoites into red blood cells. In this experiment, we immunized rabbits with 5 different antigens, including RIPR, CYRPA, RH5, or in fact the ternary complex that we made with the 3 proteins together. And we see this nice dose-dependent effect with higher antibody concentrations causing higher growth inhibition and quite big differences between these different antigens. What we also did was do this assay with different parasite genotypes, because if you're making a vaccine against malaria, you don't want to have strain-specific immunity. You want to have neutralizing antibodies that are going to be effective against all the different strains of parasite that are circulating out there. So that was comforting to see that RH5, SERPA, and RIPR antibodies are able to protect against a number of genetically diverse parasite lines. And so, however, when we compare, these are just the average values of the inhibition levels across these 4 different parasite strains. And when we compare different antibodies, we can see that in this experiment, the antibodies raised against RIPR were superior to those raised against SERPA and RH5. And kind of surprisingly, antibodies against the complex were less inhibitory to antibodies against RIPR alone. So we wanted to look a bit more specifically about, what was the mechanism of action of these antibodies raised against these proteins. So we raised some monoclonals to these proteins. And in this experiment, we had monoclonals that were raised against RIPR. And when we put these monoclonals through our growth inhibition assays, we found that 2 of these monoclonals, 1G12 and 5G6, are able to nicely neutralize parasite growth in vitro. So what we did with Wilson did these experiments, Wilson Wong, and he was able to add the antibodies together with the components of the complex, and he ran this protein mix over size exclusion chromatography, and he was able to detect peaks that represent the quaternary complex of 5G6, RIPR, SERPA, and RH5. So it appears that the mechanism of growth invasion of at least these anti-RIPR monoclonals don't involve interacting or interfering with the complex formation. So to contrast that, we had raised antibodies against this other protein called SERPA, in this case 8A7, and we found that 8A7 is actually inhibitory in these growth invasion assays. And when Wilson looked at these components by size exclusion chromatography, he wasn't able to detect the ternary or quaternary complex. He was only able to detect a peak that corresponded to the antibody bound to SERPA, which was bound to RIPR. And he was able to find all these uncomplexed components, RH5, Ripper, and the antibody itself. So this suggested that at least for this antibody, 8A7, and we also have the data that I'm not going to show, we did the same thing with an RH5 specific antibody, and it showed the same thing, that the 2AC7 antibody that we were given by Simon Draper at Oxford also appeared to interfere with the formation of this complex. So that seems to be one mechanism by which inhibitory antibodies can prevent merozoite invasion. We also worked together with Wenchang He, who was a master's student working with Ivo Mueller and Weihong Tam's lab. And Wenchang had access to this treatment to reinfection study where there was a longitudinal cohort of kids in Papua New Guinea who were followed up for 6 months and monitored every couple of weeks for malaria symptoms and parasitemia. So you can take antibody or serum from these kids at baseline, and you can measure the antibody titers to various different malaria antigens. And when Wenchang did that, he examined the titers of antibodies to either RH5, SERPA, or Ripper alone, to the binary complex, RH5 plus SERPA, SERPA plus Ripper, and to the ternary complex. And an incidence rate ratio below 1 indicates that higher antibody titers were correlated with a protection or a reduced risk of an incidence of clinical malaria. So in other words, high antibody equals lower risk of developing malaria. And so from that study, we can say that actually naturally acquired antibodies that result from an infection of malaria are associated with protection from clinical malaria. So this is, again, more evidence that this particular pathway in parasites could be targetable by vaccination. So to summarize that part of the work, we've shown you that recombinant RH5, SERPA, and Ripper form a stable ternary complex, and that this complex formation can be blocked by antibodies, and that these antibodies can inhibit merozoite invasion. So that kind of provides a rationale for complex vaccine development. So I've described that there's a stable complex that forms, and that complex formation is associated with allowing this calcium flux to happen and invasion to proceed. I've shown you that all of these components of the complex are highly conserved and that they're essential, and that they can induce strain transcending neutralizing antibodies. And work by other groups have shown that you can passively protect mice, humanized mice, from developing malaria by transferring monoclonal antibodies that are being raised against RH5 and SERPA. And work that I haven't really gone into has really detailed the specific epitopes on these different proteins that are involved in neutralizing antibody responses. So about 2 years ago, we were approached by USAID, who we'd worked with before on another vaccine project. And they asked us if we'd be interested in undertaking a sort of head-to-head comparison, a really systematic analysis of this complex of proteins as a component of a next generation malaria vaccine. So of course we said yes. And the hypothesis of this was that targeting multiple proteins, RH5, SERPA and Ripper, would be more effective than just targeting the individual proteins themselves. And that this would be a subunit vaccine approach, so it would involve manufacturing recombinant protein, and it would be done in collaboration with the University of Oxford, with Expression Biotechnologies, who were able to actually synthesize full-length conformationally intact Ripper, and IMV, who are a small startup company that make human-compatible adjuvants for vaccine development. And the funders USAID and kind of their project managers for their malaria vaccine development program, LIDOS. So the study protocol for this work basically involves making the proteins and then immunizing them in various cohorts in rats, and then taking the serum from the rats, the blood, and then making serum, and then running those through ELISAs to determine the immunogenicity of the different antigen cohorts, and then purifying the IgG from these individual animals, and then running that IgG through an in vitro growth inhibition assay. And the project was kind of organized around 3 milestones. And so the first milestone was to select the best adjuvant, which we did. And then we also started another study where we looked at the effect of different antigen doses. And then we came to the last part of the study, which was to look at antigen. So these were all done, these first two parts were done with just individual antigens. And then the third part of the study has involved combining the antigens in different arrangements. And that's what I'll talk about now. So in this experiment, we had 54 rats that were kind of divided up into 9 different cohorts. So there were rats that got the individual antigens formulated with this adjuvant only. There were rats that got the binary combinations, so RH5 plus SERPA, RH5 plus RIPR, SERPA plus RIPR. And then there were antigens that were kind of mixed together in a molar ratio, which we know in vitro, if you throw these antigens together, they just spontaneously form a complex. And so that was we did that in a molar ratio and then in a 1:1:1 ratio. And then this last cohort was just the adjuvant alone as a control. It was a 3-dose regimen and with terminal bleeds tweaks after the 3rd immunization. So we examined the functional immunogenicity of these cohorts by looking at the responses to the different antigens. And what we were able to assess was that actually it looked interesting to us because it appeared that the double immunization of RH5 and RIPR ended up giving us a much higher immunogenicity of RIPR compared with RIPR alone and a much lower immunogenicity of RH5. So it seemed like RH5 and SERPA were better as individual antigens, whereas RIPR appeared to be boosted, if you like, by combination with RH5. And that may be due to some kind of immunodominance or antigenic interference. So ELISAs are nice, but it has limitations because it doesn't actually provide any functional information on the ability of those antibodies to affect parasite invasion. And then for that, we have to go back to the growth inhibition assay. And this comparative inhibition assay measures the neutralizing activity of different cohorts of sera against parasite invasion. So we can see again that there's a nice dose-dependent effect, but this effect does not seem to correlate at all with the ELISA result. And in contrary, and this is probably easier to see in this graph here, so where we had low responses against RH5 or high responses against RIPR, we see a corresponding low growth inhibition effect. Whereas antibodies against RH5 plus RIPR seem to give higher inhibition. It appears that the triple antigen combination was good. So we see the best results with the cohorts that were given the 3 antigens, although maybe not significantly different from rats that got just the 2 RH5 and SERPA. So to summarize that, I've shown you that the RCR combination can induce high titers of neutralizing antibodies. The relatively low RIPR activity might have been due to antigen quality because this was actually a different batch of antigen from the one that we'd used previously in the other studies that I showed you. Or it may have been a dose thing because it was a much lower dose of RIPR that we used in this study compared to the previous study. Also, I haven't really gone into this, but RIPR is a hugely complicated and large protein with 10 different EGF domains. So it could be that different domains of RIPR are immunodominant, meaning that it sort of diverts the antibody response away from those neutralizing epitopes. So that result kind of posed a lot of questions. So first question that we had was that is the complex actually intact in the formulation? And we're working towards trying to work that out. We don't know whether an admixture, which is basically just adding the antigens together rather than forming the complex, is superior to just having the complex itself. And we're undertaking a kind of systematic approach towards defining neutralizing epitopes within RIPR and RH5. And Steve Scally, a postdoc in our lab, is working really hard on this. And him and Tony Triglia are also working pretty hard on trying to define other partners in this complex that might be involved in invasion. We want to determine how the antibodies that appear to inhibit invasion but don't interfere with complex formation, how do they act? And do they possibly block membrane insertion? And we also would like to know how well the in vitro assays of growth inhibition correlate with in vivo efficacy against blood stage malaria. But of course, to do that would require a clinical trial. And there has been a couple of clinical trials now with RH5 run by Simon Draper at the University of Oxford. And what they've discovered is that incredibly high concentrations of neutralizing antibodies are required to block invasion and to reduce parasite reproduction. And we don't know if this is actually possible to be delivered in a vaccine, but this is obviously a challenge that we are hoping to be able to tackle. So yeah, basically not great results from the phase 2b challenge study. At best, there was a 1-day delay to the volunteers having to be treated with antimalarial drugs. So to come back to my original question, why is it hard to make a malaria vaccine? The antigens themselves appear to be poorly immunogenic, and this is quite interesting and important in terms of vaccine development. We'd really like to understand why that is. Data from the RTSS phase 3 trials showed that antibody titers wane rapidly, and this requires a 4th booster dose. And at $10 dose, this could pose an enormous challenge for high burden countries that are also among the poorest countries in the world. As I said before, Plasmodium is a very complex creature with the extraordinary antigenic diversity and polymorphism, presenting a huge challenge to vaccine design. And also, you know, after about 50 years of researching the immunity to malaria, we don't really understand what confers immunity to malaria. And we don't have a validated correlate of protection assay that can be used to compare vaccines. So there's still a lot that we don't understand, but this provides a challenge. So we think that what we really need to get a good understanding of is what the immunology behind protective immunity to malaria is, and particularly what constitutes a vaccine-induced immune response. We really need to start looking at the interactions between cellular immunity, humoral immunity, and innate immunity. And this appears to have is directly relevant in those cases where there has been pre-exposure to malaria. It seems like even when you've got a clinical trial in non-exposed volunteers that gives a really nice protective response, when you take that vaccine out to the field and test it on people who have already had malaria, there's just really poor responses. So we really need to better understand why that is. And also, of course, children infected with malaria are usually carrying other concurrent infections as well. So what effect that has on development of a vaccine-induced response is also something that we would really like to understand. So just to conclude, I hope that I've been able to tell you about the RCR complex, RIPR, RH5, SIRPA, being critical in red blood cell invasion, and that antibodies against the components of this complex can block invasion, and that combinations of antibodies are more inhibitory than antibodies targeting single antigens, and that this could be a promising candidate for inclusion in a next generation malaria vaccine. So it just remains for me to thank everybody for listening today. And also like to thank all the members of the Cowman Lab. There's been a huge number of people who I've kind of left off this slide, but some of the key members, Alan, of course, has been extremely supportive and really encouraging. He really wants to take this research into a very translational direction. And I think before he retires, it would be lovely for him to actually get a malaria vaccine, another malaria vaccine into a clinical trial. You know, another one on top of the two that already have been developed. I really want to give a big shout out to my very good friend Jenny Thompson, who is now in retirement, who has been involved in all the way through this work that I've talked about today. Wilson Wong has been really important in the structural work. Jenny Voles for creating the conditional parasite lines. Lynn Chen, who I acknowledged in the talk for her work on structure, crystal structure, and for identification of Ripper. And then a whole bunch of other people. Ben, who I'm working with now with Steve to try to determine, you know, what's going on with this latest vaccine, whether antigens are in complex or whether they've fallen apart with the adjuvant. A whole load of work from the structural biology division, particularly Rich, who's done a massive amount of biocore work to look at the interactions between neutralizing antibodies and their epitopes. Work done in Kelly's lab for the live imaging. And of course, none of it would have been possible without Kay and her team out in Bandura. I'd also like to acknowledge the contribution of Simon's lab for providing us with proteins and antibodies, and for funds from Leidos, USAID. And yeah, that's it. I'll just hand over.
Thanks very much, Julie, for that excellent talk. And well, I think everyone listening is appreciative of that great sweep through a lot of work. So a few minutes left for questions. So we'll start with a question from Santa, which is great talk, Julie. Do we know what are the specificities of the antibodies that confer immunity to people living in endemic settings? Are they similar to the vaccine targets?
So that's a good question. In some cases, yes. So there's been quite an enormous amount of work done to try and correlate naturally acquired immunity to protection against malaria, to really tease apart responses, antibody responses to different antigens. And it appears that the different studies always seem to come up with different answers. So at this point, there is no clear antigen or set of antigens that we can really use as correlates of protection. But we can say that in a few different studies, the RH5 complex proteins have come up quite highly in screens, looking at the association of protection with the presence of antibodies to these proteins. So you know, it depends where you look. It depends also on the proteins that you use for your screen. The quality of the antigens is really important here. So there's a lot of factors to that question. So I also don't really know whether just looking at naturally acquired immunity is the way to go, because it's quite clear from the CSP work that most people don't make an immune response against sporozoites, but you can induce that response by vaccination. So, you know, you have to weigh it up.
So a question from Joe Groom. Do you investigate numbers of memory B cells generated following vaccination? If so, does this depend on the antigen target for adjuvants?
Yeah, so we haven't done that, but I know that Diana Hansen's lab and some other groups have been looking at the memory B cell response, and this is obviously something that's going to become much more important. I guess the challenge for looking at that in situ in a malaria kind of background is that they might not be in circulation. So how do you know that what you're pulling out of like a peripheral like monocyte or PBMC prep is actually what is leading to, you know, the most important immune response? Does that answer the question?
Yeah, I think that's good. Next question, can the complex be chemically cross-linked to ensure its stability following administration?
Yeah, we haven't tried that. That's actually something that Ben and Steve were suggesting that we do. I guess because of our earlier work that we did to show that the complex itself, when you compared antibody responses to the complex that was raised in rabbits compared to antibodies raised in rabbits to the individual components, it looked like the complex was less protective than the individual components themselves. So this is something that we'd like to do next. It's like a really systematic approach. So we think that potentially some of these epitopes maybe, or some of the neutralizing epitopes might be hidden in the complex. So it might not be as good to actually cross-link the complex. But that's obviously something that we're following up.
Okay, lovely wrap of current vaccine strategies. I was wondering more broadly, how many vaccines have been developed for organisms where sterilizing immunity is never developed naturally?
Well, probably all of them. I don't know that any organism where sterilizing immunity is even, desirable for a vaccine. So I think the bar's set pretty high for malaria. Yeah, I actually don't know the answer to that. Sorry.
It's an important question, really.
Yeah.
One that's been asked for many years. Jeff said, on difficulty, would malaria be the most studied pathogen with no vaccine yet produced, e.g. HIV, corona, others?
Yeah, it has been studied for a pretty long time, but that's because it's so interesting and so complicated. And so every time we seem to make some kind of breakthrough, it just seems to open up Pandora's box of questions. Yeah, I think there has to be some kind of different approach. And I know that some groups internationally are working on, strategies that aren't so kind of specific to target individual antigens. So for example, the whole parasite approach, but even that has its drawbacks in terms of like logistics, trying to get live parasites out to the middle of countries in Africa is pretty difficult.
Yeah, I think in that race HIV wins hands down, probably closely followed by coronavirus. I think malaria, that it reflects the amount of money that has not been spent on malaria compared to HIV and even coronavirus. I think our time has just run out, and Doug just left a message saying, great talk, Julie, and I totally agree with that. So thanks very much, Julie. That's been fantastic. So thanks everyone for attending the seminar, and see you next week.